talk about this later. Reinoud, if you can share? Yeah, fine. Reinoud is going to talk actually about DNA, how it is made, and what is done with it. And it's very important to understand your DNA. And that's also what Gary already mentioned. There's more as only alpha-1 antitrypsin, right? In terms of DNA, the sapina gene, there are also other genes which playing a role as well. I give the floor to Reinoud. Reinoud, go ahead. We will unmute you, Reinoud.
Yes, here
Okay,
I
go,
am, I think.
go
Thank
ahead.
you very much. Yes, I will talk about the genetics behind alpha-1. I'll walk you through how our genes control the proteins in our body and what happens when just one tiny change causes big problems. We start by looking at the different protein labels, M, Z, and S, and what they mean. Then I'll explain in 5 simple steps how a gene becomes a protein. We'll focus on what goes wrong in the Z mutation, where a single incorrect amino acid changes everything. We also look at other mutations that cause trouble. And finally, I'll share some of the developments in treatment options and emerging concepts like somatic rescue mutations. At the end, I'll leave you with a clear take-home message. Alpha-1 patients are familiar with the protein labels M, Z, S. M is good, Z is bad, and S is in between. These labels come from an older lab test called electrophoresis, where protein variants are separated in a gel and appear as different bands. You see them here, like lines on a barcode. But there are more labels. We now know of others like F, I, and null. M is for normal, S for slow, Z for very slow, F for fast, I for intermediate, and null for no protein. It doesn't show in the bands. While electrophoresis is simple and still used, it doesn't tell the full genetic story. Therefore, we have to go to the DNA, a simplified crash course in how DNA becomes a protein, from DNA to RNA to the alpha protein. Inside every human cell, there's a nucleus, the core of the cell. And in that core of the cell, there are 23 chromosomes from each parent, so 46 in all. And those chromosomes consist of the twisted ladder, a twisted ladder of DNA. This ladder has millions of steps called base pairs. And along the DNA, we find the genes, like recipes in a cookbook. Each gene contains instructions to make a specific protein. One of these genes is the SERPIN-A1 or SERPINA1 gene, located on chromosome 14. It contains over 12,000 base pairs, and here you see the base pair AT. If just one base pair is switched, say a TA here, replaces a GT, this is called a mutation, or more specifically, a single nucleotide polymorphism, or a SNP. This is the SNP. Some mutations are harmless, like replacing apples with pears in a cake recipe, but others can be harmful, like using salt instead of sugar. If both parents pass down the same base pair at a certain position, you are homozygous for that position, like in a ZZ. If the base pair is different, you are heterozygous, like MZ. The recipe has to leave the nucleus, the core of the cell, to reach the kitchen where it can be read and the ingredients can be collected. Since the DNA can't leave the nucleus, the gene must first be copied. This copy is called the messenger RNA. It's just a copy of one strand of the DNA. The instructions are written in a three-letter word, and the three-letter word is called a codon. Each codon codes for an amino acid, and that's the building block of the alpha-1 protein. The messenger RNA exits through the so-called nuclear pores from the nucleus into the cell fluid, and that's called the cytoplasm. And there the proteins are made. In the cytoplasm, you find the ribosomes, tens of millions. A ribosome is the kitchen. There the recipe is being read, codon by codon, the three-letter word. Each codon coding for one of 20 different amino acids. The amino acids are the ingredients of the protein. Here the transfer RNA fetches the amino acid and puts it on the appropriate place on the messenger RNA. And then the protein is released and added to the other, the amino acid is released and added to the previous amino acids, forming a chain of amino acids, and together they form the protein. Sometimes, as in the recipe where the salt replaces the sugar, there's a wrong amino acid. Maybe it's this yellow one, and that causes a lot of problems. The protein is the dish. It's the result of what is happening in the ribosomes. This is the alpha-1 protein. There are 394 amino acids linked together and folded in an intricate way. Even if just one amino acid in the protein is wrong, and you see here at this place in the protein, the green square, that's the place where the Z mutation is. It can mess up how the protein folds. It's like bending a paper origami the wrong way. The shape of the protein is absolutely critical to its function. In our case, trapping elastase. Proteins are like tiny molecular machines, just like any tool. Their shape determines what it can do. If the shape is altered, even slightly, as in the case of the Z mutation, the protein may lose its function, work incorrectly, or cause disease. In this square, you see the glutamic acid, and it is replaced by lysine. There's just a tiny difference between those two molecules, and that is the cause of all trouble. The trouble is that the protein becomes sticky, and it causing it to clump together. And that's what we call polymerization. Other broken proteins, they lump together And this is the most well-known cause of alpha-1. But there are more troublemakers. With the SERPINA1 gene covering 12,000 positions on the DNA, you can imagine that more mutations are possible. By now, researchers have listed about 500 mutations. 200 of these are associated with some kind of deficiency, ranging from mild to very severe. Here you see in red a newly discovered mutation called Chillicothe, because it was discovered in a patient from Chillicothe, Ohio. On the right, you see a novel Z-type mutation in blue next to the pink one, which is the Z mutation. And this one is called Z Little Rock because it was discovered in a patient from Little Rock, Arkansas. Every year, new mutations are discovered. This is a list from 2 years ago of 22 new mutations discovered by the University of Florida. We don't know the precise effect of most of the mutations yet. Each mutation can alter folding, secretion, or function in slightly different ways, which is why deeper genotyping matters. Here you see the Chillicothe mutation, and here the Z Little Rock. What are the new developments targeting these mutations? Well, at the level of the gene, we see that there's a novel treatment developed called base editing. It's replacing just one base pair, and this is, for instance, the company called Beam is working on it. There's also prime editing. Prime editing is replacing chunks of DNA, or sometimes even the whole gene. Prime Medicine is a company working on this kind of therapy. Then we also have companies working on changing the messenger RNA instead of the DNA itself. Wave Life Sciences and Takeda are working on that. And finally, this company, Biomarin, and we will shortly hear from them, they work on stabilizing the protein and preventing polymerization. So there are different developments which are very interesting and promising. Then there are some emerging concepts. Recently, a recent discovery is that the people with alpha-1 deficiency get older. Some of their liver cells develop new genetic changes in the SERPINA1 gene. These spontaneous changes in liver cells are called somatic rescue mutations and may offer natural protection. This shows that in alpha-1, the liver may adapt by developing special mutations that reduce harm. And it's a study by Martin Corina and Campbell. Another rather recent development is the focus on patients with multiple mutations, the so-called compound heterozygotes. These mutations can cause increased severity of the disease. The impact of the mutations depend on the degree of function loss, the severity of folding issues, and the degree of polymerization due to both folding issues and secretion defects. take-home message. Well, the most important one is categorizing variants as Z or null or S is far too simplistic. We need full subpoena genotyping, especially in atypical cases and in MZ individuals with systemic symptoms. The gene has to be checked for many more known disease-causing mutations than up till now. iGene is a European company testing specifically for alpha-1. You can find it on our website. This has direct implications for patient care. It allows personalized risk assessment, careful monitoring for complications beyond liver and lungs, and ensures that future gene editing strategies take variant-specific behavior into account. The message is clear. In alpha-1 antitrypsin deficiency, personalized insights are vital, and comprehensive genotyping is essential. The Alpha-1 MZ Foundation is driving this mission forward, but to fully understand the impact of this gene mutation, we need your support and funding now. Thank you.
Thank you very much, Reinoud. It was an excellent presentation. I will do my presentation very, very short with an eye on the time to give Jenny a little bit more time. So I will do only one slide. If you can stop presenting, Reinoud, then I will share my thing here. Hope everybody can see this. This is the last slide actually of my presentation about emerging therapies. So I will do it very quickly and very short. There are a lot of different solutions which are on actually developing a medication, which Reinoud is telling us, right? From changing DNA to changing RNA, inhibiting RNA, etc., etc. So there are about, what is it, 10, well, about 15 companies working on a solution now for alpha-1, right? This slide is actually a summary of that all. All the different parties, pharmaceutical companies are in different phases. Some are preclinical, some are already in phase 3. But if you look at the right side at the blue lines over there, you see how much time it takes. Besides the sheer amount of money it takes, the time is also quite relevant. So if you're in phase 2, right, you still need about 2 to 3 years to complete phase 2 typically. Phase 3 may take a little bit longer, and then even if you look at approval, that may even also take a year. So currently Takeda is, I think, the furthest. They're in phase 3 right now, and I think they're in phase 3 already for a year roughly. So they have a couple of years to go, so they may be closer to a solution where you actually inhibit actually the Z. So you inhibit the Z polymerization, right? Belsiran is the second one over there, which is discontinued. So here you see also that not every medication is going to make it to the market. They discontinued in phase 2 because they had too much issues, right? And with this, actually, I'm going to give the floor to Jenny over here, which is Biomarin, which is currently in phase 1. And you can look at these tables and the rest of the presentation maybe later on. I will share that later on. I will stop sharing and then Jenny can start her presentation on Biomarin.